All right, we are recording. You should be able to see my screen. I wish somebody was here to verify that, but I don't. I'll just have to move forward without it. So here we go. Set a timer for 50 minutes.
50 minutes starting now.
All right, that'll keep us on track. So before I want to start, I want to really reiterate this fact that if you're feeling overwhelmed, don't be, and let me help you. The beginning of the semester is almost always the hardest because not only are you learning the new material, but you're just learning how the course works, right? Like, where do I find the labs? How do I turn in labs? Where do I find the resources? All those things you have to deal with. And it's so much, and it's already overwhelming. But then add to that all these issues we've had with weather, that makes things much worse. So if you're feeling overwhelmed, I know it's easier said than done, but try not to be. And if you need help, just like I said, let me know. I'm here to help. Given all the issues with weather and stuff like that, I'm going to be pretty flexible as far as due dates are concerned. So yeah, just let me know. Um, anyway, moving forward, exam one. We've speaking of the weather, we keep having to push things back. Exam one, we've pushed back again. Now, if you remember, the original plan was for us to take it in person. You're supposed to bring in your computers and take it in person. But we're so far behind now that I don't want to waste a lecture day taking an exam. So when I'm done with this lecture, I'm going to reopen, or not reopen. I'm going to open exam one. It'll be open today, and you'll have, it'll close this Sunday. Just take it on your own time whenever you want. It's almost all we've almost covered basically everything that's going to be on it, which is mostly chapters 1 and chapters 2. There will be a little bit from chapter 3, and I'll cover that once we get started with chapter 3. But yeah, other than the fact that we're not meeting in person, everything's the same, right? It's closed book. You only have 50 minutes to take it. There's 50 questions, about 50 questions. And if you need more time to take it, you need to be in touch with me now, so we can make arrangements for us to meet online so I can proctor your exam. If I can ensure it's a closed book exam, then I don't mind you taking longer to take it. So anyway, that's that. Oh, which reminds me, I have one really great, a couple of them, but some students from the online version of this course, and they were watching these announcements. So I just really want to quickly remind anybody watching, I mean, not these announcements, but these lectures. I want to remind anybody watching um, these announcements are the logistics I'm talking about. That's almost entirely for 101-02, the in-person students. So the material is the same. So it's good for you to learn from the material that I'm teaching in the lectures. But as far as logistics are concerned, like due dates and things like that, this is for the most part only in reference to the in-person students. Anyway, speaking of in-person students, we will have the lab tomorrow. I see no issue. There shouldn't be any weather issues. So make sure you're there on time. I know I just talked about don't be overwhelmed and things like that. And I am being pretty loose because I want to help you not feel overwhelmed. But one of the few things that I am strict with is being on time for lab. So if you're not present or if you're not on time for lab, the doors will be locked and you'll just have to get the data from someone else and you won't get full credit. And that's not a good thing, right? But it's just not fair to the rest of the students if you come in late because then you don't know what you're doing, right? So anyway, just keep that in lab. Keep that in mind. Another thing I want to mention is we have no pre-lab due this week. So that's some good news, right? Because here you have this exam that's due Sunday. So that's probably weighing on your mind. Plus the fact that, again, Lab 4 is going to be in person. So we actually are going to do it. So you'll have to deal with that because that'll be due on Friday as usual. So yeah, no pre-lab this week. I won't waste any time explaining why today. I'll explain it during lab tomorrow. But yeah, no pre-lab due this week, as in this Friday, right? The only thing you need to turn in this Friday will be Lab 4, which we're going to do tomorrow. As always, I'll remind you attendance is mandatory and participation is graded. It's the same stuff as usual. Just send me an email throughout the lecture. I'm going to say a series of words, write those words down, take a screenshot, and then send me in the email with that list of words and the accompanying screenshots. And this part that a lot of people seem to forget, ask one question about the material covered in the lecture. Yeah, and I think that's it. So let's just say the first word for attendance is going to be grade. Grade is the first word for attendance. So if you screenshot that, I should see a picture where I have circled the word graded. So, all right, let's jump into it. We are now covering chapter 3. And like I just mentioned previously in that last slide, there's going to be very little, if any, questions about this on the exam. And there's a reason for that. So even though obviously cells are important, as you learned in chapter 1, cells are the unit of life, right? When we talk about life, that is the fundamental unit of life. So cells are important. The thing is, for the first half of the semester, you're going to be learning all about cells past this chapter, right? You're going to learn how cells reproduce. You're going to learn how cells convert energy from glucose to ATP. You're going to learn how, some cells convert sunlight energy into glucose and things like that. So you're going to learn how cells work from here towards the end. So the way I see this, chapter 3 is simply an introduction to things that you're going to learn later. So again, there's hardly going to be any questions about this on the exam. And for that reason, and for the fact that I'm recording this and nobody's actually with me, so you have the ability to pause this, slow it down, do whatever you need to. So I'm going to go through this kind of quickly. So there we go. Chapter 3, structure, cell structure, and function is broken down in these 6 main bullet points you can see here. We're going to look at how cells are studied. We're going to compare prokaryotic versus eukaryotic cells. Then we're going to really dig in a little bit deeper into eukaryotic cells. Then we'll talk about the cell membrane, passive transport, and active transport. So let's jump into it right now. The learning objectives for this first portion are, by the end of it, you should be able to describe the roles of cells and organisms, compare and contrast light microscopy and electron microscopy, and summarize the cell theory. So again, as far as the exam is concerned, this stuff is not extremely important. But so with that in mind, let's jump into it. First of all, microscopy. I'm not going to ask you any questions about this. We're not even going to be doing any of this in lab. Now, if any of you feel strongly about it, there is one free lab that we have scheduled at the end of the semester. So I'm happy to fill that in with a microscopy lab. So if you want to do that, let me know. Anyway, let's move forward. Not that I'm going to ask you this on the exam, hence the big X I'm putting through right here. But there's different types of microscopy. There's light microscopy. There's dissecting, which is what you see here on the left. There's dissecting microscopes, which you have on the right, and an electron microscope, which is not pictured. Let's talk about cell theory. Your textbook says that the unified cell theory states that all living things are composed of one or more cells, and that the cell is the basic unit of life, and that all new life arises from existing cells. So this is one of those things that probably will be on the exam, and I said there wasn't going to be much. But in this case, this is actually something you learned in chapter 1. I already taught you about the unified cell theory. I just didn't give it the name, unified cell theory. And if you do have any questions about it on the exam, it's not the name. You don't need to know the name, the title, unified cell theory. You just need to know that all life consists of cells, right? That's the important thing here. Same thing here. I'm not going to ask you about these two gentlemen, but your book mentions them, so do I. Leeuwenhoek and Robert Hooke, both of them were basically pioneers in microscopy and cell theory. Leeuwenhoek was the first to observe bacteria and protozoa, and Robert Hooke coined the term cell. So the word cell came from that guy. And both are credited to be the first 2 scientists to discover microorganisms. So again, as far as the exam is concerned, it's a very unimportant slide. That first bullet point is important, but not necessarily that you need to associate. You don't need to memorize what the unified cell theory is by name. But again, you just need to know that all living things come from cells, and all the stuff we already talked about. So let's jump into 3.2, comparing prokaryotic and eukaryotic cells. There might be a couple questions on this. The learning objectives of this section are, by the end, you should be able to name examples of prokaryotic and eukaryotic organisms. You should be able to compare and contrast prokaryotic cells versus eukaryotic cells. And you should be able to describe relative size differences in these different types of cells. So before we compare and contrast, let's talk about some things that are the same, whether we're talking about prokaryotic or eukaryotic. First of all cells have a plasma membrane. And we talked a little bit about this when we talked about biological molecules. You learned about lipids, and one particular type of lipid that you learned about was a phospholipid. So that is where this comes from. And it sounds like somebody joined me. So if you did, welcome, but I'm going to keep moving. I had said previously when I started recording that I'm going to go through this quickly because we're a little bit behind. And as far as the exam is concerned, actually, as far as understanding stuff throughout the semester, this chapter is relatively unimportant, relatively. So that's why I'm going to go through it quickly. Anyway, that's the plasma membrane. The cytoplasm is the jelly-like region where all the other cellular components are found. So basically, it's the stuff inside the the cell, right? So on the outside of the cell, you have the plasma membrane. On the inside, you have everything else that's basically suspended in the cytoplasm. We also have DNA, which I'm not going to ask you about, for this exam. Well, not much, because you already learned about DNA in the previous chapter where we talked about biological molecules. You learned about polynucleotides and how it's the genetic material, what DNA is. So that stuff will be a little bit more important later in the semester. This is, again, just an introduction to it. Same thing with ribosomes. I'm probably not going to ask you any questions about ribosomes on this first exam, because that's going to be something we talk about in detail not much later, but later in the semester. So any questions about this slide? All right. Sorry, again, I'm going to go quick. So don't, feel free, whoever's here, to stop me or slow me down or whatever it may be. So let's talk about prokaryotic cells. There's basically two types, bacteria and archaea. The two types, first of all, there's three domains, and this is something we touched on, I think, in chapter 1 when we, started at domain and then goes down to kingdom, phylum, class, so on and so forth. So that'll be, again, important later in the semester, but this is still just an introduction. But anyway, domain is the largest category when we're classifying life, when we're talking about taxonomy. And again, as you can see here, it's broken down into three domains. And prokaryotic cells, again, they make up these two, which are two of the domains. And the other one, which we're going to talk about later today, is the eukaryotes, right? So those are the three different domains of life: eukaryotic, archaea, and bacteria. And again, archaea and bacteria are prokaryotic. So far, that's the only thing you need to know about prokaryotes, right? That they include bacteria and archaea. Another thing you need to know is that they are simple single-celled organisms, right? They're unicellular. They don't have a nucleus. They don't have any other membrane-bound organelles, as a matter of fact. So throughout this chapter, once we get past the prokaryotes and we're talking about eukaryotes and we're talking about all the parts of the cells, those are going to be organelles. So again, prokaryotic cells don't have organelles, at least not membrane-bound organelles. And once again, even though I'll be teaching you about a lot of organelles today, there's not going to be many questions on them on the exam, because we're getting those important organelles, at least important as far as academics are concerned, and as far as the exams are concerned. We'll be talking about those again later in the semester. And a great example of that, again, is DNA, right? DNA you've been introduced to. We're really going to cover that later in the semester. I'm teaching out of the textbook. It's bringing it up again, so I'm bringing it up again. The DNA is the central part of the cell in the darker region called the nucleoid. So if we're talking about prokaryotic cells, not eukaryotic cells, but prokaryotic cells, bacteria and such, they don't have a nucleus. They have something called a nucleoid. But that is something I'm not going to ask on this exam. So any questions about this slide? All right. And really quickly, too, I've been updated about disability compliance. So just to explain what's on the screen here, there's a picture of a prokaryotic cell that has, it's labeled, the parts are labeled. And just so you know, you do not need to know any of those parts. So yes, the picture is there. It does label the parts, but I'm not going to ask you about the prokaryotic cell parts in the exam. All right, so that's it for prokaryotic cells. Eukaryotic cells, unlike prokaryotic cells, do have organelles. Organelles are membrane-bound compartments or sacs that have specialized functions. In a sense, you can almost think of a cell like a factory, and the organelles are different machines that have different jobs within that factory. So yeah, that's what an organelle is. I'm not going to ask you what an organelle is. Later in the semester, you're going to become familiar with different types of organelles, and you will need to know about them at that point. But again, this is just an introduction. You might need to know the size difference between prokaryotes and eukaryotes, but not necessarily these details, right? You can see here it's pretty specific. A prokaryote is 0.1 to 5.0 micrometers. A eukaryote is 10 to 100 micrometers. You definitely won't need to know those numbers. If anything, you just need to know that prokaryotes are smaller than eukaryotes. Eukaryotes are larger than prokaryotes. And again, to describe the screen, what we have is a picture here from the textbook that shows different types of things, just so you could reference their size, and compare their sizes, right? So the very left of the spectrum, we have the very smallest thing that they have, which is an atom, which is 0.1 nanometers. And then on the very right, we have an adult female, which they have as a 1 meter. And it also breaks down which parts you can see on the electron microscope, which is going to be the things towards the left, things you can see on with your naked eye, which are things that are going to be seen on the right. And of course, some overlapping with that with the light microscope, which is the stuff in the middle. But for the exam, this image is not important. Again, if anything, you just need to know that eukaryotic cells are larger than prokaryotic cells. But I'm definitely not going to ask you any questions. For example, what's larger, a frog egg or a human egg? Of course, you can see here that it's a frog egg, but that's not a question that will be asked on the exam. So do you have any questions about this slide? All right. The next word for attendance, I'm circling it. Might be a little bit hard to see, but it is egg. Egg is the next word for attendance. All right. Really quickly, before I go to the next cell, I'm going to back out of here and make sure that I'm still recording. And make sure that's not what that noise was. Good. That's not. It was Marley joining me. Good. All right. So we're still recording. We're still in business. And get back to it. All right. So Marley, since you're here, just to let you know, if you want to, you can just go ahead and send, do like you usually would do, write down those attendance words. Since you're here with me, you don't need to do the screenshots. Just send me the attendance words. Include one question at the end. And if you get a chance, you might want to go back and watch the beginning of, what little bit you missed. You missed the announcements. So that would be helpful for you to watch that. But anyway, let's get back to it. Let's talk a little bit more about eukaryotic cells. And I will say this too. Almost everything we talk about in this semester, like I said, later, we're really going to dig into the different processes of cells. And almost entirely, it's going to be about eukaryotic cells. So you've been introduced to prokaryotic cells. But when you learn about respiration and photosynthesis and mitosis and meiosis and transcription and translation, all these things are basically going to be eukaryotic cells. Right? The more complicated, the larger. And we are eukaryotic organisms, so it kind of makes sense that that's what we're focusing on. By the end of this section, you should be able to describe the structure of plant and animal cells. And again, that will not be on this exam because later that will be important. Or when we talk about photosynthesis, you're going to need to know about plant cells. When we talk about respiration, you're going to need to know about animal cells and, again, plant cells, because plant cells also do respiration. Anyway, you should be able to state the role of the plasma membrane. You should be able to summarize the functions of major cell organelles and describe cytoskeleton and extracellular matrix. So what we have here is a picture that has all the parts of the cells labeled and described. So it's a picture of a cell with little arrows pointing to it. So for example, the nucleus is subdivided into the nuclear envelope and chromatid and nucleus, the nucleolus, peroxisome, so on and so forth. So as far as the exam is concerned, again, this is not necessarily important. We're going to talk about all these things that are currently on the screen, but we're going to talk about them separately. And for the most part, you're not going to be graded on them. So let's talk about the plasma membrane first, which makes sense because, again, the cell, that's what's on the outside of the cell. Everything else is on the inside. So it makes sense that, we would talk about basically what defines the cell. As we mentioned in chapter 2, when we talked about the different biological molecules, you already learned about phospholipids. Remember, that was one of the types of lipids you learned about. Those phospholipids make up something called a phospholipid bilayer. So you need to know that, right? That's what the cell membrane is made up of, phospholipid bilayer. That'll be slightly important even for tomorrow's lab because we're working with diffusion and osmosis, and you're going to have these, well, other than the Petri dish experiment, you're going to have these little fake cells. And chemically and physically, they do kind of act like a cell membrane. So they let some things in. They don't let other things in. But anyway, that's the phospholipid bilayer. You need to know about that. And its main function, other than to just keep things inside the cell, is to also regulate what comes in and out. And again, that'll be more important later in the semester when we start talking about photosynthesis and respiration, because the ability to regulate what comes in and out is actually going to be very important later. So any questions about this slide? All right, and before I move forward, again, to describe what's on the screen here, there's a picture from the textbook that shows a cross-section of a phospholipid bilayer. It also has parts labeled. But again, I'm not going to ask you about this on the exam. and we will talk about these individual parts that are important later. So I'm not going to go through discuss or explaining everything that's on this slide because again, we'll come back to these important parts. All right, cytoplasm. We already mentioned this. We'll talk about it again. This is the contents between the membrane and the nuclear envelope. That's one way of putting it. That's the way the textbook puts it. I would say it's just the stuff inside the cell membrane, right? It's just the stuff that makes up the cell. Granted, there are going to be parts within that stuff, such as the nucleus, right? And the organelles. But basically, it's just the stuff that everything is in. It's a gel-like substance, or it contains a gel-like substance called cytosol. That cytosol also contains cytoskeletons and various chemicals. But if anything, again, I'm not going to go this deep. You already know your cells are mostly water. The textbook in this chapter says 70 to 80%, but I'll remind you for exam 1, since you haven't taken it yet, you need to know 70 to 95%, right? That's the number that you need to know that we learned in chapter 1. So don't let that confuse you. I'm just teaching out of the textbook here. And many metabolic reactions take place here. And we're going to learn about those reactions later in the semester. So any questions so far? All right, the cytoskeleton is very important for cells, very unimportant for the exam. I'm probably not going to have any questions about it, but I will explain how it works. And it'll be helpful to know how it works when you learn stuff later. But even this isn't something, even if you didn't understand this or you forgot this, you could still have a pretty good understanding of the stuff you're going to learn later. So again, this makes it relatively less important. The cytoskeleton is a network of protein fibers. So that would be a good question if I was to ask something, but I don't think there are any questions about that. But yeah, what's a cytoskeleton made up of? Proteins. What's it do? It helps maintain the shape of the cell. It secures certain organelles in specific positions. So again, like I said, you could think of a cell almost like a factory, and these organelles are different machines that do different jobs. Well, sometimes you need those certain machines to be in specific places, and the cytoskeleton helps achieve that. It allows the cytoplasm and vesicles to move within the cell. I haven't even told you what a vesicle is yet, but we'll talk about that later. Again, relatively unimportant as far as the exam is concerned and as far as understanding concepts later in the chapter, later in the semester. And then finally, the cytoskeleton enables unicellular organisms to move independently, and we'll come back to that. Even though there will be no test questions on it, we will come back to that. Any questions about this slide? All right. There's 3 types of cytoskeletons. I will not ask you about any of them, but they're in the textbook, so really quickly, here they are. First of all, there's microfilaments. These are also known as actin filaments. These are the smallest. So the one I'm describing here, we're going from smallest to largest. These are the things that move cellular components. They maintain the structure of the microvilli, which is folded cell membranes. The idea of a folded membrane will be slightly important later when we talk about photosynthesis and respiration, but we'll come back to that concept when it happens. For now, this is just an introduction. Microfilaments are very common in muscle cells, and they are responsible for muscle cell contraction. As is the case with anything I'm teaching you here, I'll remind you, feel free to look up any of this from a separate source if you want to look up more into it. Write a paper about it if you want. Don't forget your sources. So any questions about microfilaments? All right, the next one is intermediate filaments, which is a great name since there's only 3 types and we're going from smallest to largest. The middle one is called the intermediate filaments. It's an intermediate diameter. And its function is that it maintains the cell shape. It helps anchor organelles. And as far as its structure is concerned, it is made of, some of it is made of keratin. Again, not very important for the exam. So any questions about intermediate filaments? Okay, really quickly, microtubules. These are the largest, the thickest. These are hollow tubes. They actually rapidly dissolve and reform. They also help guide organelle movement, and they will move chromosomes. So microtubules will be very important later in the semester. But when you learn it, when they are important, that's when I'm going to teach you more about it. So for now, again, this is just an introduction to these concepts. You'll learn more about them later. But again, not only do they move chromosomes, which will be the important part later, but also basically trivial for us, they also move flagella and cilia, which help cells move. Before we move forward, actually, are there any questions about microtubules? All right. So before we move forward, I'll explain the images on the screen. What we have is two different images. One of them it basically shows what intermediate filaments look like. The other one shows what microfilaments and microtubules look like. None of this is important. It's just there so you can see it, but you'll never get tested where I show you an image and ask you, which one of these is intermediate, which one of them is microfilaments, which one of them is microtubules. So if there's no questions on that, we'll move forward. Again, I brought this up earlier, so in the textbooks, bringing it up. So here it is, but I'm not going to test you on it. Flagella, cilia. Flagella, basically the long hair-like structures that extend from the plasma membrane that move the cell. Right? Not all cells have them, but when they do, there's usually one or very few of them. Right? So, for example, sperm cells have flagella. That's how they swim around. Cilia. These are much shorter. They're basically like little tiny hair-like structures that are around the entire surface of the plasma membrane, and it's also used to help move cells, just to help to move them around. Yeah, and when they are present, and again, not all cells have them, but when they do, there's a lot of them. So if you were to compare and contrast these two, which again you're not going to have to, but if you were, flagella is larger, longer, and there's fewer of them. Cilia, smaller, and there's more of them. So any questions about that side? Okay. The endomembrane system. Again, this will be a little bit more important later in the semester when we talk about how DNA works and how DNA replicates and how we go from DNA to RNA to proteins. But for now, this is just the introduction. These are membranes and organelles that work together to modify, package, transport lipids and proteins. We won't even talk about the lipids. So this is probably the first and last time I'm mentioning that. But yeah, when you have these lipid molecules in your cells, this is where it's all made. But again, later in the semester, we will definitely talk about how proteins are made when we go from DNA to RNA to proteins. But that will be important then. The whole system includes the nuclear envelope, which we're going to talk a lot about later, lysosomes, which we probably won't talk about at all, vesicles, which we might talk about a little, the endoplasmic reticulum, which we'll talk a little bit about later, and the Golgi apparatus, which again, we'll talk a little bit about later. So again, to reiterate, none of this should be on exam one. This is an introduction because you're going to learn more about it later in the semester. But anyway, let's break it down and talk about the different parts. The first one we're going to talk about is the nucleus, and there's an image on the screen that shows a nucleus, and it has an image. Excuse me. This image is labeled, so it shows where well, I will describe this one. This one's a little bit more important, even though, again, you will not get a question where I show you this picture and ask you to label it. But anyway, just in case, let's work on the inside out. The very inside part is called the nucleolus, and I'll talk about what that is later. And then outside of, and this is all inside the nucleus, by the way. So the outside of the nucleolus, you have the chromatin, which is the DNA, the chromosomes that we've talked about before. That is still inside the nucleus. You also have the nucleoplasm, which I won't even really get into, but it's just like the same stuff that's in a cell, except it's in the nucleus. So it's that gel-like substance, except it's in the nucleus, so it's called a nucleoplasm. Then finally, as we start to get out of the nucleus, there's these little holes in the nucleus, which will be important later in the semester. Those are called nuclear pores. They allow things to come in and out of the nucleus. And again, later in the semester, that'd be important. Of course, when I talk about there being holes in the nucleus, what I'm talking about is the nuclear envelope, right? So there's the nuclear envelope, which is basically like the cell membrane of the nucleus. And again, it has those holes in it. And then outside, now we're talking about outside of the nucleus, you have the endoplasmic reticulum, which we'll talk about later, and it's shortened to be the ER. Yeah, and that describes the entire picture. But anyway, with that out of the way, let's talk about the words that are on the screen. The nucleus. It is the most prominent organelle. That will not be a test question, but now you know. You probably already know because I've already hinted at it, but the nucleolus is where the cell's DNA is, right, in the form of chromatin. Now, later in the semester, you are going to learn that there are some exceptions. There are other places where cells have DNA, right? So your main DNA is in your nucleus, but as you're going to learn later, your mitochondria has its own genome, and for plants, they're chloroplasts. They have their own DNA, but we'll talk about that later. Anyway, the endomembrane system, the nucleus is what we're talking about now. This directs the synthesis of ribosomes and proteins, and that is what's going to be important later in the semester. Again, right now, this is just an introduction to the concept, which is one of the reasons why I'm going so fast. So, are there any questions about the nucleus? All right, let's talk about the nuclear envelope, which I basically described when I was talking about the image, but this is a double membrane. So it's a phospholipid. Well, not that this is important, but I do want to make sure you understand. And when I say it's not important, I mean, I'm not going to test you on it. But when I say it's a double membrane, when I think about your cell, right, your cell is a membrane, right? You have the cell membrane, which is one phospholipid bilayer, like we discussed earlier. The nuclear envelope is actually a double membrane. And that would be important if we were studying things deeper. So that is, as far as life is concerned, that's an important fact. But as far as the exam is concerned and understanding concepts later in the semester, that is not important, right? You could completely brain dump that and still be able to learn the things that I need to teach you later. As I already mentioned, it is the outermost portion of the nucleus, and it has holes in it that controls what kind of things can come in and out of it, much like your cell membrane does. So once again, before I ask if there's any questions about the nuclear envelope, I'll remind you, this will not be on the exam. The important stuff about the nuclear envelope will be discussed later in the semester. So are there any questions about the nuclear envelope? All right, let's talk about the chromatin. Again, we're going to have a whole long discussion about this later in the semester. So for now, this is just the introduction. I'm going to breeze through it very quickly. Chromatin is basically chromosomes, right? This is what chromosomes are made up of, is chromatin. And it's about 50-50. It's made up of DNA and protein. You already learned a little bit about DNA in chapter 2. You're going to learn a lot about it later in the semester. And we'll also even talk about the protein that helps make up the chromatin. And that'll be important later because these proteins have different functions as all proteins do. But as you already learned from chapter 2, when we're talking about DNA, this is the hereditary material. Not that you need to know this, but it is linear in eukaryotes. So you'll often see a picture, and I know if you can't see this, I'll try to describe it. I'm drawing. I got this long, squiggly looking thing, and that's a chromosome. It's linear, right? It's just one big, long thing. And I'm only bringing that up again because the textbook did. So when we're talking about prokaryotes, which we're not, but if we were talking about prokaryotes, they have just basically one chromosome, and it's a gigantic circle. Not that you need to know that. Anyway, another thing that'll be, I don't know, slightly interesting, maybe important, because you might get one question on this later in the semester. So I will bring this up again later in the semester. Species have specific numbers. So, for example, humans, by definition, unless there's something they have a chromosomal abnormality, they have 46 chromosomes. So you have, unless there's something wrong, 46 chromosomes. And I have, unless something's wrong, 46 chromosomes. Not that you need to know. Well, you will need to know that later, but not for this exam. So again, I don't know. In my opinion, that's slightly interesting. So if you want to for extra credit, you could look up some of your favorite species and look up how many chromosomes do they have. I don't know. A certain palm tree, how many chromosomes does that have? Certain fish, whatever it is that you're into, feel free to look it up for extra credit. Don't send that on the attendance email. Just do that on some other time. Anyway, any questions about chromatin? Okay, the nucleolus. I've already described it. Here it is again. This is the dark area inside of a nucleus. And when I say that, I mean, you'll see we'll have a lab later in the semester where, unfortunately, we look at pictures of microscope slides. So you won't actually get to see them in person, but you'll get to see that dark spot. And that's what the nucleolus is. But as far as the exam is concerned, as far as understanding concepts later in the semester, nucleolus is not very important. This is where we find ribosomes, and we will talk about ribosomes later. Excuse me. Let me rephrase that. This is where ribosomes are made. But that part, as a matter of fact, won't even be important. When we talk about ribosomes, we're going to be talking about what they do outside of the nucleus. Not that it matters, but there's this picture that I've already described that has the nucleus with all the different things labeled. One of the things that's not labeled on here is these little red dots, right? So if you look if you could see it, we've got this endoplasmic reticulum, which I already described, and they have little red dots on it. Those are ribosomes, and ribosomes will be important later. But again, the fact that they're made in the nucleolus, completely unimportant for any exam this semester and for understanding material later in the semester. So any questions about this slide? All right. So next is the ER, the endoplasmic reticulum. I've already mentioned it a few times. Again, not important for the exam. It'll be slightly important later when we talk about how proteins are made. But even then, when we talk about how proteins are made, we're really going to focus in on that process of going from DNA to RNA and then from going to RNA to protein. Even then, we're not even going to focus that much on the fact of where it's happening, which is the ER, the endoplasmic reticulum. So anyway, really quickly, since it's in the textbook, I'm gonna give it to you. These are interconnected membranous, interconnected and the membranous. Sorry, there should be an and right there. They modify proteins. They not only do they modify them, well, we're going to do that later. And they also synthesize lipids. So again, later in the semester, that middle bullet point of modifying proteins, that's when we'll come back and talk about this. But again, even then, we are so focused on the processes, the steps of going from RNA to protein that we barely. I mean, we'll talk about the fact that it has ribosomes, but we won't probably, discuss much about the fact that this is happening in the ER. So, any questions about this slide? All right. The Golgi apparatus, also called the Golgi body. This is a series of flattened membranous sacs. This is where your cells sort, package, tag proteins and lipids. And they also, so again, really quickly, just to remind you, this will not be on exam 1. And this is not important at this level of biology because later in the semester, when we talk about how we make proteins, that's like the end of the discussion for us. We don't even talk about the fact that after those proteins are made, they have to be sorted, they have to be packaged, they have to be tagged, right? And we don't even talk about how we make lipids, right? So again, as far as the exam is concerned, as far as this semester is concerned, the Golgi apparatus is not very important. But anyway, the receiving side of the Golgi apparatus faces the endoplasmic reticulum, which makes sense because if you remember, that's the part that makes the proteins. So it's going to make those proteins and send them to the Golgi apparatus where it receives it. And then after it's done sorting and packaging and tagging these things, it releases it out into the cell membrane, right? Or faces the cell membrane. So whatever these things that it produced, if they're going to be used by the cell, there you go. They're in the cell now. Or sometimes your cell is going to export them, in which case they will leave the cell membrane. But anyway, again, for the exam, this is very unimportant slide. There's a picture to describe it. It's a scanning electron microscope picture of a Golgi apparatus. There's something called a transface and a cisface. These aren't terms I don't even think that your textbook has. I don't even think this is from your textbook. But it's a good picture of what a Golgi apparatus looks like. You can see for scale that line right there is 200 nanometers. So you can guess that's probably like what? 1, 2, 4, 6, maybe 800 nanometers across. But again, I'm putting a big X to this because this is very unimportant for the exam. The whole Golgi apparatus is very unimportant for us, much less a picture of it. Are there any questions about the Golgi apparatus? All right. Next thing we'll talk about is lysosomes. Again, this is just as unimportant as the rest of the stuff. Very important for life. Very unimportant as far as exams is concerned and as far as understanding things later in the semester. But again, I'm teaching out of the textbook. So here we go. Lysosomes are basically the garbage disposal. So they have these digestive enzymes. We haven't really talked a lot about enzymes yet, but I have introduced you to the concept. Those are proteins, right? Proteins that basically make certain chemical reactions happen. And in this case, the chemical reactions they're making happen is digestion, right? Certain enzymes break down certain things. So all those macromolecules that we talked about in the last chapter, the proteins, carbohydrates, lipids, all that, these are the things that break them down. So when you eat, you eat that food and you need to break it down because you want to release the energy that's stored in the chemical bonds of that food, but you also want the building blocks. You want those monomers so you can build new things. And this is what lysosomes do. And they also happen to destroy disease-causing organisms. Really quickly before we move forward, I'll try to explain this picture that's on the slide. What we have here is something called a macrophage. Now I think about it, I might not even need this picture because, again, this whole thing is very unimportant, but I think this is, for us. But what we have here is this bacteria has been basically engulfed by this cell, which is a macrophage, and we don't even need to talk about that. But anyway, it's engulfed this bacterium, because that's singular. So now that bacterium is inside this vesicle, and maybe it's a bad one. So this vesicle is basically breaking it down into its pieces, right? So it's trapped in the vesicle. The lysosome comes up and it forms, it merges with the vesicle, and that's where it breaks down the bacteria to its parts. So again, I'm only describing that for ADA compliance. This is actually quite unimportant as far as the exam and the semester is concerned. Any questions about this slide? All right, the next word for attendance is below. I mean, literally, that's the word. I'm not saying look below and you'll see the picture. The next word for attendance is the word below. Below. All right, vesicles and vacuoles. So you can see here, I mentioned, sorry, I backed up the slide here. I'm back at the lysosome slide. Here, like I said, this vesicle, it's basically just what happens when something comes inside. We'll talk about that later. We'll talk about phagocytosis. That's something you'll learn later. When something comes into a cell, it's just surrounded by the cell membrane, a little bubble of cell membrane. And in this picture, that's what the vesicle is. And the vesicle itself doesn't do any digestion, but again, the lysosome merges because the lysosomes also have their own little membrane. So they merge with the vesicle's membrane and they become one and they do their job. But anyway, that's what a vesicle is, right? So that's what this slide is. Vesicles and vacuoles. Like I already mentioned, they are membrane-bound sacs. So that picture that you saw previously when I was talking about the vesicle, it's just like a little bubble. It's like its own little tiny cell membrane. It's used for storage and transport, right? So in that case, it kind of stored up that bacteria and transported it to the lysosome. If anything, and I'm probably not going to ask you this either, vacuoles are larger than vesicles. So if you would need to know the difference between vesicles and vacuoles, there you go. Vacuoles are larger than vesicles. And these are the things that we use to break down macromolecules. But regardless of whether they're talking about a vesicle or vacuole, well, let's just focus with the vesicles, I guess. They fuse with membranes. So that, then again, if you could see, you would see that picture of that last slide, which was that membrane just kind of butted in and it had its own little tiny little membrane. And that's what happens. And then actually, I will just back it up. So that in that case, right, something has come into the cell, right? So now we have this little vesicle that has its own little membrane. But what you're not seeing here in this picture, sometimes things need to exit cells. And we'll talk about that later in the semester too. So if there was an arrow here, what would happen is it'd basically be the opposite of what you see on the left. It would kind of merge with the cell and then bloop, the thing would plop out basically. So anyway, that's vesicles and vacuoles. Any questions about that? All right. Again, ribosomes, very important later in the semester. Very important for understanding how things work later in the semester. No questions about it on the exam, but these are the things that make up proteins. You'll need to know all about them later, but not yet. They're made up of two parts, a large subunit and a small subunit. We'll talk about it later. Mitochondria. This is another big one. We'll have almost a whole chapter based on this. So when we learn about respiration, you're going to learn how mitochondria work. A lot of times you hear people say the mitochondria is the powerhouse of the cell because, like I mentioned earlier, when you eat, right, your body takes that food, those molecules, and it breaks them down to release the energy that's held in those covalent bonds of that food, those molecules. And it uses that to create something called ATP, which you're going to learn about and need to know about later. But ATP is basically just an energy source. And again, later that'll be important. Mitochondria also happen to have a double membrane. That will also happen to be important later in the semester, but not yet. Like I mentioned earlier when I talked about DNA, they have their own DNA. So you have your DNA, which is a mixture, your 46 chromosomes that I mentioned earlier, that's made up of yours and your mom and your dad's DNA. That's where it came from. But then you had this mitochondria has its own DNA. And not that you need to know this, but sperm don't have mitochondria. So when a sperm fertilizes an egg, it's only the egg that has the mitochondria. What that means is your mitochondria, the DNA of that mitochondria is 100% from your mom. There's no dad influence, and that came from her mom without any dad influence, which came from her mom, so on and so forth. Back for millions or, tens of thousands of years, however long, that's a direct lineage from the mom side. Any questions about the mitochondria? All right, peroxisomes. Once again, very unimportant. So unimportant that for us, academically unimportant. We won't even talk about them later. This is the first and last time I'm going to talk about peroxisomes. You don't need to understand how they work to understand any of the stuff later in the semester. But these things happen to have some single membranes, and what they do is they break down fatty acids and amino acids. So once again, we're just talking about, again, this idea of digestion, right? Those macromolecules that you learned about in the last chapter. This is just some of the ways they're broken down. Another thing they do is detoxify many poisons. But again, for the exam, for this semester, peroxisomes are very unimportant. Any questions about peroxisomes? All right. Really quickly, a little comparison and contrasting between animal cells versus plant cells. Plant cells do not have centrioles, they don't have centrosomes, and they don't have lysosomes. That's not important, but it's in the textbook. I mean, academically, that's not important for us, but it's in the textbook, so I'm including it. Meanwhile, animal cells do have those things. Plant cells have cell walls. Animal cells don't. Plant cells have chloroplasts. That's where photosynthesis happens. Animal cells do not. Both plant and animal cells have something called a plasmodesmata, which we'll talk about later. And even then, it's not going to be academically important. Same thing with plastids. And, well, looks like this is a little bit messed up too. So sorry, this shouldn't say this. The animal cells do not have a large central vacuole, but plant cells do. This is where they store their water, and you'll learn about that later too. If you've ever seen a plant that's wilted, that's why. It's because it's dehydrated, so it doesn't have that large central vacuole that's filled with water that's keeping it turgid. But again, that'll be later in the semester, not important now. So any questions about this slide? All right, the cell wall. Again, plant cells have it. So we're talking about plant cells here. We're not talking about animal cells. It's also worth noting that we're not even discussing fungi, and some fungi do have cell walls, but we're not even bringing up that discussion, and you can look into it if you want for extra credit. But the cell wall is external to the plasma membrane. So we already talked about what the plasma membrane is, right? That's the outside of the cell. And plant cells, just like every other cell, have that. But again, outside of that, they have this additional layer, and that's the cell wall. It protects the cell. It gives it support. It gives it shape. As you learn in chapter 2, when we're learning about carbohydrates, we said one of those is cellulose, and that's exactly what the cell wall is made up of, cellulose. And again, like I mentioned a couple seconds ago, fungi also have cell walls, so do protists. And even bacteria, which is completely prokaryotic and not eukaryotic, even they have their own version of cell walls. But again, we're just focused here on plant cell walls in the slide. And again, academically, not important. Well, there'll be a couple times it comes up. Again, when we talk about cellular reproduction, obviously, if we're going to reproduce a cell, we're also going to need to discuss how we make the cell wall, but that'll be important later in the semester. Any questions about this slide? All right, we've got less than 2.5 minutes left. I'm going to fly through this. Chloroplast, again, we're going to discuss this in greater detail later in the semester when you learn about photosynthesis. So for right now, this is just your introduction. I'm not going to test you on the first exam about this. This is where photosynthesis happens. It happens to be double membrane. That'll be important later in the semester. But for now, there's your introduction. And really quickly to describe the image. It's an image. It's another labeled image of an organelle. So you can see this bean-looking shape. It looks like a bean. The color and the shape of it looks like a bean. It kind of has a cross section. And you can see inside of it. First of all, you can see that it has an outer membrane and an inner membrane. Like I mentioned, it's double membrane. And there's a space in between that, and that'll be important later in the semester. There's these things called thylakoids, and granum and stroma. All of that will be important later in the semester. But even then, the image is not necessarily important because I'm not going to give you a picture of an image and ask you to, label it like you see here. Central vacuole, like I mentioned earlier, plant cells have them, animal cells don't. This takes up most of the cell. So plant cell is mostly the vacuole. Like I mentioned earlier, this is where the water is. So it regulates water concentration. And this is what keeps plants rigid and not wilted. Not important academically for us. So I'm going to move forward. Same thing with extracellular matrix. For us, this is academically unimportant. Again, obviously for life, all these things are important, but I'm not going to ask you any questions about the extracellular matrix. It happens to be made up of glycoproteins, which I haven't even mentioned yet, and I'm not going to test you on that. But as the name suggests, it's proteins that have glycogen on them, right? Basically, it's a basic description. They also have collagen that's secreted from also made up of collagen that's secreted from the cells. This is what holds cells together to make tissue. So as you learn in chapter 1, right, if we start small, the levels of organization and get larger. We have atoms that make up molecules, and those molecules make up macromolecules, and those macromolecules make up organelles. Organelles make up cells, and then cells together make up tissue. And this is the stuff that keeps those cells together. That also allows them to communicate. And really quickly, again, just to describe what's on the screen here, there's a picture of the extracellular matrix that's labeled. You can see the phospholipid membrane. You can see the inside of the cell. You can see the outside of the cell. But again, for the exam, extracellular matrix is academically unimportant. And that picture is even more so academically unimportant because I'm not going to show you a picture of that and ask you to label it. So it's been 50 minutes. When we come back, we'll talk about extracellular junctions. The last word for attendance is going to be GAP, G-A-P. So go ahead and send me the email. And again, if you're with me right now, all you need to do is send the email with the list of words and one question, right? One question about the material covered. And of course, if you're watching the video at a later date, go ahead and send the screenshots and a list of words and the question. And again, for Marley, who's here, you don't have to, but I really highly recommend once I post this video, at least go back and watch the announcements because those are pretty important. So that's all I have. Any questions? All right. If there's no questions, I'm going to quit recording. And I'm going to see you all on, well, tomorrow. So that's it. And have a great day. I'm trying to quit recording. It's not working for me here. Very strange. All right. Let me just try to hang up then.